as he says, I'm going to be talking to you about the twilight zone, or twilight reefs, really. That's originally what this zone was called back in the 1970s. So I'm going to talk to you a little bit about that. The picture here is actually one of my favourite Gorgonians, which is what I actually specialise in. It's called Anella. Now, so a little bit of a run through, so it's going to let me, we're having some technological marvels happening this morning. Okay, so the plan for this morning is I'm going to tell you about twilight reefs, or what is also called mesophotic coral ecosystems, or MCEs. I'm going to let you know where they are found, how we go and look at them and explore them, what is actually there, and then I'm going to walk you through some of the trips that Ocean First classrooms funded us to go and do, which is fantastic. And so you can see here on the side there, there's just me down there at 135 meters that was taken by Richard Pyle, who I believe is online, and there I am collecting Gorgonians. And as you can see there, there's this reef, this wall, actually, and it's completely dominated by Gorgonian corals, which is my research. Okay, so the shallow coral reefs, people generally associate those with scleractinian, or hard corals. And here's a lovely picture of a hard coral here out in Indonesia. And they have zooxanthellae, or single-cell algae, that are associated with them. So they require photosynthesis in order to survive. And that's what we're used to hearing when we talk about coral reefs. And they're absolutely delightful, of course. So the reason why we know probably a great deal more of that, because they're more accessible to us. So you can see we've got a little diver here coming down on the screen. And so most of the research of the marine realm has been conducted by scuba diving. And scuba diving is largely where you're blowing bubbles. Every time you breathe out, you've got lots and lots of bubbles. And that's the difference between rebreathers and open circuit scuba. I'll touch a little bit more on that in a minute. But the exploration that has generally happened in the marine environment has happened in the first 50 meters of water. And this is also largely due to our physiological limitations. If you start going past the 50-meter realm, then the oxygen that you breathe actually becomes toxic to the system. Now, if you're going to want to actually start going deeper, you're going to need a sub, right? Or more to the point, if you're going to want to start going deeper, the submarines, they generally cost like up to about $39,000 a day. If you're going to be spending that kind of money, you're going for grants and all that kind of stuff, you're going to be wanting to go a lot deeper. Okay, and so people go off to the abyssal plains and all these really fascinating places. And there you are, there's the Pisces, there's a submarine, but you've got this small little porthole in there, and you can't actually see what's going out. It's like seeing the moon through the telescope. If you're actually an astronaut, you're on the moon, you can see what's happening. But if you're just looking at through this small space, you're not actually going to see very much. And that's what it's like when you're looking through the window of a submarine. So there's this whole area here which nobody really knows about, and that's what we call twilight reefs, or mesophotic coral ecosystems, which was called by NOAA back in, I think, 2006 or 2008. And they're generally characterized by in a low light environment. So that's where photosynthesis is actually decreasing and being taken over by organisms that do not need to photosynthesize. And the way we do that is we use rebreathers, okay? And so rebreathers, as I mentioned earlier, for the open circuit is where you actually start blowing bubbles. Well, a rebreather essentially recycles your air, and you are actually able, it actually enables you to recycle that air through a system and replenish it with the oxygen that you need. This is actually a very kind of, it can be a complicated procedure, and there's Richard Pyle and my friends have actually explained it in much greater detail, and you'll find that information on the internet very freely. But bottom line, this is what we use. It extends our bottom time. It tailor-makes our oxygen to our physiological requirements, and it enables us to do the research that we do. It's a much smaller and much more compact and efficient system. And as we're actually doing the diving, we're collecting lots of data. The data also goes towards the Poseidon systems in order to actually contribute towards refining this rebreather. But also we're collecting data, environmental data, temperature and salinity and all these other different things. So every time I collect a sea fan or the guys collect a fish, we know exactly what environment that animal was taken at, and then we can use that as part of our scientific research. And we're constantly streamlining this process. Okay, so the biogeography, where do we find these twilight reefs? Well, they're circumtropic, okay? And these little golden stars here are actually where even myself or the group that we're involved with have actually been over the past 2 decades. And so here's the red stars of where other people have been. Now, if you consider the actual huge expanse of space here, there's a lot of areas that we just have not gone and explored. And there's this understudied realm that every time we go down there, we find something new. It's absolutely fascinating. Okay, so here's some example of the fishes that have been collected over the past 2 decades or more. These are new species to science that have been collected from depths way beyond 50 meters to 150 meters. This goes on and on and on. And what you also find is when you start going in to the twilight zone, you will actually find that there's less than 15% species overlap in those species, in these species that you actually find there. So this actually suggests that we're dealing with a completely different ecosystem, and this brings it to endemism. So what you see in this picture here are every single fish is endemic. Endemic, that means that you will only find them in the Hawaiian Islands. You have 9 species of fishes here. For endemism, not everyone's familiar with this actual term. As I say, it's unique and specific to or location to the region. It's also, this means it has an important role in the origin of biodiversity. We want to work out why these species are endemic. And so they can also be key conservation indicator species for those locations and those locations themselves. Again, they're harboring new species and, well, usually new species to us, but also specific species to that region. So the point is every time we go down into the twilight zone or on those twilight reefs, we actually are finding large levels of endemism. So twilight reefs are generally what is often publicized as a beautiful 100% coral cover of the zooxanthellate corals, of these hard corals. And this is a picture taken by her of Maui Island in Hawaii, about 67 meters. But what we're actually finding throughout the Indo-Pacific, but also the tropical Pacific, the circumtropic, and the Atlantic as well, are these reefs. And these are generally dominated by Gorgonian sea fans, also black corals and other octocorals and some sponges. And so this is what I'm actually focusing on, and this is what my research focuses on, and I'll be touching on today. So when you look at coral biodiversity, the actual scleractinian corals here, the hermatipic corals, they are the ones that the reef-building corals actually constitute 14% of the overall global coral biodiversity. And what the large majority of the remainder of these are octocorals, which is 64%. So really, even though the hard corals that we see on the reefs are actually, there's no dispute, they are extremely important. In terms of global biodiversity, it's actually dominated by octocorals, and those are largely concerned with Gorgonians. So here are just some of the new species that I keep finding every time I go down onto the twilight reefs. Every time you go down, we always see something new. And typically, it's even more exciting when I go beyond 140 meters. It's like someone presses a button and I'm dealing with a completely different, just sort of realm of species. It's really quite amazing. And this is, yes, this is, it just goes on. It's like being a child in a candy store. So just to give you an example of, it's very rudimentary because the work on the Gorgonians, There's very few of us actually doing it, and in the Indo-Pacific there only seems to be myself. So here, just a very quick example, these green spots here are just Gorgonians that are actually found in the shallows, and these blue spots here are those found in the twilight zone, okay? Now, this is a very common pattern that people are seeing, that if you start off in the west of the Pacific, you've got high biodiversity, but as you go over to the east of the Pacific in the shallow reefs, this biodiversity actually decreases. But as you see down here, as you go across the east of the tropical Pacific, these blue spots remain. That means that the biodiversity, even though it is actually diminished in the shallows, it stays the same pretty much on the twilight reefs. Well, why is that? And so part of what we're doing is actually trying to work out the origins of the biodiversity and why that seems to maintain itself in the deep, where it actually is extremely less in the shallows. So what's a Gorgonian octocoral? Okay, I'm just going to walk you through this very quickly. Gorgonians, octocorals, their tentacles are multiples of 8, which is very, very simple. When you look at the scleractinian corals that you see on the reef, it's basically tentacles, they're 6 or multiples of 6, 6, 12, 18, 24. Also on Gorgonians, you actually have these pinnate tentacles, and they increase the surface area for them to get nutrients and so on and so forth. So it's very, very simple. That's the difference. Now, they have a high global abundance and diversity, as mentioned before. You can find them down to the abyssal plains. You can find them also in the Antarctica, mangroves, just all over the place, highly diverse. They also can harbour conservation flagship species, and in some areas actually are flagship species. So here we've got these pygmy seahorses. They're absolutely delightful. This one here is definitely a new species to science. And as I say, the deeper you go, the more of this sort of stuff that you find. It's absolutely fantastic. There's a high morphological variability, which is you can often look at what looks like a different species, but actually it's just the same species doing different stuff in a different environment. And so that makes it quite difficult to identify things. They're also suspension feeders, so they stick themselves out into the water column in order to collect all the creatures and what have you, and the marine snow, which is often bacterial dominated, in order to eat and survive. Now, there's many ways to be a Gorgonian. There's gorgeous, gorgeous things. You've got these deep spiral type shapes here. You've got these fans here. You've got whips, and you've also got something here called brarium, which I was in complete denial that was a Gorgonian at all, because it's actually encrusting. This is a zooxanthellate one. So there's many ways to be this kind of creature. Now, I'm just going to talk to you about why these are important in the twilight zone, or on twilight reefs. So here we've got our little diver there, blowing bubbles. He's going down to 50 meters, right? And if you notice there, I've got the temperature. This is 30 degrees centigrade, okay? And then you've got your little submarine, and you've got these twilight reef zones. And again, when you go down to your submarine, this is like 2 degrees centigrade. Now, the twilight reefs usually, where we start seeing really profound differences are 100 meters plus, especially 140, as I said before. But the temperature really starts to dramatically drop. So what I'm seeing is what we call Gorgonian bathymetric polyphyly. Oh my God, look at all those syllables. Bathymetric is another way of saying depth. Polyphyly is like what we think are actually very similar, closely related groups are actually very different. And I'm looking at loads of different groups here and species within these groups to try and find out why they seem to be so different. So to put this in a different context, I'm going to give you an example. So here you've got something what we call the Isididae, or bamboo corals. Bamboos, because they look like a bamboo. You've got this jointed axes. This is part of how we identify them, different axes structure. And so in the shallows, you've got this axis, the brown stuff is the protein. Proteinaceous is called Gorgonin. That's where the Gorgonians get their name from, and it allows them to sort of flow in the water column. But in the deep, why do they need to be so flexible? Okay, so I'm looking at what we call structure-function relationships. So you've got these two different species here that are supposed to be related, but actually aren't, but look the same. So Gorgonian polyphyly, again, we're going to look here, the shallow water ones, you've got the fibers, the protein fibers here are actually very, very baggy. And so, as I say, it allows them to flow around in the high water flow, the shallow waters. And yet in the deep, you actually got these fibers are very close together. And this kind of close together, I mean, they don't really need to be flexible in the deep water. So why are they retaining this signature? Why are they retaining this kind of feature? Again, structure-function relationships. But in the twilight reefs, I'm seeing lots of intermediates that may give us answers to how these species have evolved, and some have gone shallow and some have gone deep. And so this is an ongoing concern, ongoing research that I'm looking at. It's very difficult to elaborate more because there's so much more science that I haven't done yet. Some of these species bioluminesce, you know, it's like, wow, how do they do that? So many interesting things. So as I said, I'm going to touch a little bit on the twilight reefs of Caine, Ant Atoll, and Ponape. And this is what Ocean First classrooms actually helped us fund to get there. Now, here's Brian here collecting some fish. Brian and Richard, you're looking at decades of experience, rebreather diving and collecting fish. So they're just down there, they make it look so easy. But I know when other people have tried to copy them, you know, they spend their whole time trying to get one fish, where Brian and Richard, they've just got their bucket full of fish. They've been doing this for so many years. It's a dream to watch. So where are we actually going here? Just in Micronesia. There we are, sort of right in the middle of everything. Caine, Ant Atoll, and Ponape. There are some of the sites that we went on a research expedition, as I say, funded by Ocean First. Now, here we are. There's Richard Pyle, Dr. Richard Pyle, Brian Green there, and myself. Just the three of us who went down there and decided to find out what we could see, start running some experiments, and finding new species for science. Now, here's just an example of one of our dive profiles down to 150 meters. You can see here it takes very little time to actually get down to that depth. This red line here is our depth. And then we spend a few minutes just down there gathering what we can find as fast as possible, and then slowly making our way up, doing the decompression, but still collecting material on the way up, but documenting what we find, and we're often setting down data loggers, temperature, salinity, that kind of thing. We're characterizing this environment. But also I wanted to highlight to you that the rebreathers that we use, here is the gas. I mean, there's the oxygen, and there's the diluent, sorry, and there's the oxygen, and we're not actually using that much. If we were on open circuit, we'd be just draining that completely. Right, so a little bit of an example. People got questions, we can come back to that, as I say, we're only limited on time. But I wanted to highlight temperature. So the thing is with the twilight reefs, or mesophotic coral ecosystems, is they're characterized by huge gradients in environmental variables. Okay, often people talk about light. We're going to be losing a lot of light. But also one of the key things is temperature. Now, temperature is obviously associated with the sunlight that we're actually going to be losing as you go deeper and deeper. But on the mid-oceanic islands and atolls, you've got very clear water, so the sun can penetrate a lot deeper. So temperature, however, is also affected by deep water upwellings, submarine groundwater discharge, and other internal wave structures that you have in the oceans. So there's a lot of colder water that's coming in and affecting the reefs. Now, this is a very rudimentary bit of data that we collected. So as you go down, going backwards, sorry about that. We got 30 meters as usually. This is actually from Ponape and the conglomeration of all the data that we collected there. You've got around 29, 30 degrees centigrade from 30 meters. Okay, and so then when you get down to about 80 meters, you've got these thermoclines here. We get this, so as you're descending, we really feel those thermoclines. Okay, so we're down to about 24 there. And these thermoclines are actually associated with very discrete or distinct faunal differences. All of a sudden, the fish that you're actually going to be seeing and the Gorgonians and the sponges, the creatures that you're seeing, they shift. You have these abundance and diversity shifts. By this point, a lot of the scleractinians have gone. And then you've got around 120 meters, you've got another kind of thermocline here down to 16 degrees centigrade, and then you've got it creeping down again to about 50 meters here, and this is what it sounds like. Hear it? I'm not sure if you can hear that. Yeah, you obviously heard some of it. It is really painful. It's like, if you sit there and you can imagine, you get these ice cream heads or brain freezes, like, oh my God, you know, you're swimming into this and you can see the thermocline, you're swimming into this, oh my God. And these boys are dressed in board shorts, you know what I mean? Like, oh, heavens above. But I tell you, it makes it even more exciting when you're down there. You're just like, right, you're completely focused on whatever goes on. I mean, I'll be quite frank, it's my absolute favorite place to be. Now, so here we go, Gorgonians. This is what I find. I get down there. This is what I'm seeing. I just bed down. Usually I'm going behind these chaps, and so I can see what they're doing, but not getting in the way of fish collecting. Because, of course, the fish are much more volatile, the Gorgonians. You can just sit there and you collect them. So I'm going to walk you through very briefly what happens, how I collect them. Here, every time I see a Gorgonian that I want to collect, which is very simple, I just go down there, there's tons of them. I take a picture of the sea fan coral. I take a picture of my paddle, which tells me all the information about the environment and my rebreather. I take either the coral or a small sample. I have a pre-labeled bag there, and I pop it into a larger bag and move on to the next one. I also look around me to see where everyone else is. As you can see, my lights here, my antenna, so my hands are as free as possible. So here is my paddle. I mentioned about that. It tells me all about my dive information. So every time I see a coral, I also look at this paddle. So I'm always constantly know what my oxygen is, what my time to the surface is, what my temperature is, what the time of the dive is, what my depth is, what my battery's doing, what my gases are doing. So constant awareness. That's always what I want when I go down there, constant awareness. Because otherwise you kind of lose your peace of mind. You know where you are, you know what you're doing, you know what the corals, the environment they're in, and you can use that data and you can refine your protocol the whole time. Okay, other things I'm doing, I'm collecting for coral mucus. I'm looking to see what kind of relationship the Gorgonians are actually having with their microbes, because Gorgonians have a reduced gut, very small gut. And so if they've got a very, very small gut, how are they able to eat properly? My hypothesis is that they're gardening bacteria. This is not unknown in the marine realm and also in the natural history realm of a terrestrial environment, plants and things like that. So I'm looking at that, looking at their transcriptomics, looking at seeing if they've got actual relationships, genetic relationships, genomic relationships in how they're able to survive. Are they supporting each other? Again, if people have questions about that, you know, please bring them up. You're welcome to email me as well. It's kind of a big subject area. I could waffle on for ages. I could generally waffle on for ages. So what happens as we start to ascend, as you can see here, Rich, he's kind of looking longingly at the boat on the surface, you know, because we're down there for a very, very long time, you know, up to sometimes 5, 6, 7, maybe even 8 hours. But luckily, there's still lots of Gorgonians. I keep sampling up the different depth ranges, and then I can do scientific comparisons on what we can find. What are the patterns? It's very, very, you know, to me it's extremely important when you're actually down there. Unlike a sub, when you're actually down there, you see cryptic species. You see species and animals and interactions that you would not be able to see through the lens of a camera, through the eye of an AUV or an ROV or a sub. You're actually there in situ with the creatures. And this is also the difference between what we call bulk sampling. You know, if you go down, you drop down a quick grab and something like that, you're not seeing the entirety of what they are. So actually being down there and actually seeing the creatures, you can then start asking and answering questions much more thoroughly. So this is what we do on decompression, which is good, keeps us off the street. And here, as due to the funding of Ocean First Classrooms, are able to start doing a field aquaria, and I'm working with the Waikiki Aquarium, being able to keep these amazing creatures at the temperature that they are, and the light that they're used to. So here, it's very simple. As you can see, in a hut environment, we've got this old sort of bucket affair and this chiller, and this is actually in the guys' room. They've got their AC on full blast. This is obviously not unpacking, there's no AC whatsoever, there's hot. But we're actually able to keep these creatures and transport them back and actually start finding out about how they're able to survive in these environments and the evolutionary mechanisms of change. Also, when you get to the surface, this is actually when the majority of the time is actually spent around the field. It's specimen processing. So here I am, I'm collecting for different ways, looking at their reproduction, looking at their genetics, their genomics, and I'm tallying them up there, as you can see, with the images that I've actually got. And also, sometimes we run out of ethanols. I have to go down and use rum. They've used these in past expeditions back in the 17 and 1800s. So if it works for them, it still works now. It still preserves my specimen. Of course, everyone thinks I have an alcohol problem, but actually the specimens get all my alcohol. The other thing that's extremely important to us when we go out is what we're actually doing now is community outreach. And here's just a very simple image of myself with Sonia Rowley, which is actually the chief of Caine Atoll. Caine, there's people on Ponape, which is only 27 kilometers away, that never get to this atoll, or very, very rarely. Others can get over there a little more regularly. Those that are associated with the Conservation Society of Ponape, who are really helpful to us. These people, there are around 80 people on the islets of the Caine atoll, and they are so enthusiastic. And I've never actually seen reefs so clean in my life. They were very accommodating to us. Someone had donated a generator to them a few years ago, and they put out a big old wooden table. They kept that generator running all night, and I was surrounded by the local community finding out what we had found. So as you can see, those are my bags of Gorgonians, and we're just there looking at them. Look how excited they are. We started playing, like, counting how many brittle stars are on the Gorgonians. And I just wanted to highlight this young lady here. She stayed with me until after midnight. She must have been about 4 or 5, I don't know. We didn't share the same language, but she followed and copied everything I did. It was quite amazing, and they're genuinely interested in what we found, and we shared as much as we possibly could. I believe that we cannot keep what we have without giving it away, and that's what we do actively. And obviously within the paradigm of Ocean First Classroom. So again, this is a reminder of what Caine Atoll looks like. From the surface, this is at sunrise of the lagoon. This is the clean environment in which they live. This is involved over, I think they've been out there for maybe up to 3,000 years, and they do not put their rubbish in the environment. This is a natural way of being able to circulate and live in closely with nature. But twilight reefs are not all as beautiful and as fantastic as we think they are. And this is not to their fault. It's obviously because of our impact. And the impact is often associated with, unfortunately, more continental regions. So this is a reef, a deep reef, around 90 meters, I think, in the Philippines. A lot of human activity on the surface. And these guys here, there's Brian and Richard trying to catch fish. You can see the sediment is building up. You know, by the time they try to catch the fish, you know, there's this huge fog because of all the built-up area that's on the surface. We also see our human signature really down deep. This is beyond 140 meters, where I really start seeing new species to science. However, they are impacted here by fishing lines and cans. I've been out on deep ocean expeditions out in the Southern Ocean to the Louisville Seamount chain, and we still see our signature of trash and fishing lines. So we need to be aware of this. We could be losing these reefs before we've even discovered what's down there. So I wanted to actually share with you guys a video, but I'm not sure this is going to work. So what I'll do is I'll let Caine actually share this video with you after the webinar, or we can at least try in a bit if we have time. But often there's questions as to how I got into this line of work. So I'll try and answer a couple of those before we move on to the questions that have been popping up on the screen. My parents started diving when I was 3 in 1976, and there's me. We need a bit of a scale bar. There's me and my elder brother Giles, tiny little thing where they threw me in neoprene, and they would go off diving, and we'd watch them from the surface, and dive down and try and catch them, and it would be all very exciting. My father is a wreck hunter, and he would find all these wrecks and took us with him. All 4 of my mother as well. She's a very accomplished rebreather diver. And that's how we were raised. It was my first dive. I was 11 years old, over 30 years ago. Wow. And I can remember it to this day. The colours were amazing, and so you get a sense of peace I never ever get on the surface. It's ultimately fascinating, and my absolute dream was to be able to sort of marry diving science with marine biology, and I'm fascinated with evolutionary mechanisms, as you may have guessed. And so I spent a large part of time on this boat, which is that my parents owned and ran from England. I'm actually diving right now. There's my mother, and that's my bedroom up in the wheelhouse. So that was a lot of my childhood. So this is how I actually came to do the diving that I've done. I'm very, very privileged to have had decades of experience of diving around the world in all temperatures, many, many different depths, and be able to put that back into the community now is a real gift. So I just want to have my acknowledgements to obviously Ocean First Class Education. I think I've been saying classroom. Ocean First Education that enabled us to go out to Caine Atoll and Ponape. We're actually setting up experiments. We actually set up experiments that are actually new to science, where I'm putting Gorgonians in different places and testing how they respond to that environment. And we can certainly keep you posted how those go. And here again are just some of my collaborators and my research team, which is Richard Paul, Brian Green, and then we have Gwen, the Waikiki Aquarium, Wilbur, and all the people at Ponape and Caine Atoll. So I think with that, I'll be happy to take any questions. I do apologise for the technical difficulties. It's probably me. I'm not massively technically savvy, much better under the water. So yeah, there we go.
All right, thank you, Sonia. That was a very insightful talk. I am fascinated about what you have been up to. So if you have questions, please write them in the chat section there, and we'll try and curate them for Sonia. I picked up a few during the talk from some of you. But first, I was really interested in how many new species of fish in octocoral have you discovered? A rough number might be enough.
Oh my God. There's just so many. I mean, when we went to the Philippines on those 2 trips, I had at least I sit there and say 20, but every time I look under the microscope, I see something new. It's a really hard number to say because there's far less research actually done on Gorgonians. With fishes, there's a lot more research, a number of, at least 2 decades. I'm going to be quite frank, I actually don't know the answer, and I don't think anybody does. I mean, because every time we go down, it's just new. Everything's new.
That's amazing.
Yeah.
So with you were showing the endemism curve from west to east. Do you think that's something to do with sea level rise and fall over geological timescales?
Yes. So especially in the last 2 to 4 million years, the Quaternary and throughout the Holocene, you were dealing with over 30 glacial sea level changes, rises and falls. You've got these stark sort of water gets locked up in the glaciers, and that makes the overall global oceans a lot shallower. Shallower up to over 130 meters shallower. And then when the actual environment starts to increase in temperature, then that water that's locked up in the glaciers then melts, and that increases our sea level up to what we're basically seeing now. So you've got these oscillations of sea level cycles going up and down and up and down and up and down. And what happens, as I was explaining towards this east-west attenuation, it's quite a big thing to get into, but when you're dealing with oceanic islands and atolls, they're much more susceptible to these variations in sea level change. You know, of 150 meters, that's a huge kind of variance in sea level going up and down, up and down, up and down over the millennia. Now, every time you've got that sea level that goes back up again, the shallow water species need to be repopulated in some way. Now, if you're really far away from the nearest landmass, then that makes it quite difficult for species to sort of get over that expanse of ocean to then repopulate this habitat. So that's a large, that's largely what to do with why we're seeing so less species diversity in the shallow. You've got this constant up and down, this cycle of the sea level rise and the temperatures, which takes away the habitat and then puts it back again, takes away, puts it back, takes away, puts it back. But when you go down into the twilight depths of 130, 140 meters, we're starting to see we do see a completely different signature of organisms. And because they've not been subject to this constant sea level cycle change, they're enabled to be persistent over geological time. And actually, Gorgonians are older in the geological and paleontological record than Scleractinia. So they've actually evolved coping mechanisms over hundreds, thousands, millions of years, over and above many, many other species that are down there. So every time we go down, we have a historical signature. We really are going down and seeing in your face a natural history museum. And so yes, those sea level cycles have huge impact on what we see in the biodiversity. And there's a lot of very active research, especially on fish, with regards to sea level cycles, habitat persistence. Rich and Brian and other collaborators are actually, publishing work with regards to the glacial sea level, the effects of glacial sea level changes over time. I think that answers that question.
Yeah, for sure. That's a great answer. I suspected it was something to do with sea level rise and fall and how far organisms have to swim, especially in the level phase. So Emily L. asked a great science question, which is kind of always the bane of our existence. How do you decide where to collect samples?
How do we decide? Well,
yeah.
well, there's a number of different ways. Often you look in these various environments, especially when you look further and further out into the mid-ocean, you're like, oh my goodness, how is anything, as we've just discussed, how is it going to get there? How does it survive? What is there? Okay. Also, we've got like historical information. I love to go and visit all sorts of different museums to learn what species they have there, but also to help them, identify certain things as well. There's often these black boxes of unidentified species there, but also you get these really interesting, very old records, 100 years ago plus, and these really fascinating expeditions where they found stuff and they've deposited it in the museum, and that's it. And of course, I'm looking at those and I'm like, wow, I want to go back there and see what else is there, because if I see this species, then this is probably associated with it, and you've got all these different patterns. So the patterns of biodiversity that you see, that I'm more and more discovering, and also the different the physical oceanography, I'm looking at the different patterns, not just me, we're looking at the different patterns that you see around the globe, particularly the Indo-Pacific, because it's so old and so fascinating. You're like, right, okay, so if you've got this ocean current at this particular depth range, and it's oscillating around here, and it's probably going to go around this location, then if we go and explore down there we're probably likely going to find X, Y, Z, or we're actually not sure what we're going to be finding, but there's a lot of nutrients in that area due to the physical oceanography or the geology. So we're likely going to start being able to answer certain hypotheses. So in short, which is really difficult to say in short, there's a number of different aspects of the ocean, of the geology and the biology in which enables us to pinpoint where we would like to go next.
That's, yeah, I mean, scientists always have a trouble with trying to figure out where to sample, how often to sample. Assuming it's difficult in the mesophotic zone to replicate what you would do at the surface level, but I found it interesting that you use museums and museum records to help you decide. I think it's an amazing aspect of how valuable museums can be to current day science. So that's really cool that you're actively engaged in digging through the archives and finding things that were found hundreds of years ago and using them to your advantage. A great question from Mariana. Have you detected any indicators of climate change impacts down there?
Yes. Now, when we actually go down, we see wave-cut ledges. I've talked about these thermoclines. They're typically associated with these ledges, and they are showing us climate change, you know, where reefs were. There's fascinating things. I've talked about the glacial cycles, and there's some wonderful, very interesting research which correlates very much with what we see. It's like these kind of punctuated marks of where the water's been locked up in the ice, and then you've got this gradual kind of temperature change, and then all of a sudden you get this kind of release of water, and then that changes the actual sea level. And of course, this is where the scaractinians are extremely important to what we see, because they're creating these ledges. So we see a historical signature of where reefs once were, but these are in an often punctuated way. So we do see the signature of climate change of times past, but the climate change of now, as opposed to what we're seeing anthropogenically accelerated. Yeah, we do see that. Well, I mean, as I mentioned earlier, we see the anthropogenic signature of trash and so on and so forth. But we do probably, I've seen in certain areas, more in the shallow area of the twilight reef, around the 40 to 60, 80 meter realm, you do see bleaching. You can see bleaching, and it does make you wonder. I've actually got lots of, down in Ponape and down there right now, we've got a lot of temperature loggers down there every 10 meters, so we can actually see the oscillations and see how that changes. But this is an ongoing research concern. It's certainly not the only one, and one of the experiments that we've got down there at the moment is to actually see how these organisms respond to different temperature changes and how that's enabled them to survive or not over the millennia. So this is an ongoing concern. One of the things we have to be aware of is, especially on the mid-oceanic islands and atolls, is the atolls are so low-lying. What the indigenous populations are now seeing is the sea level is rising to such an extent that they're losing their land, they're losing their ability to grow crops, and they're losing their ability to sort of have a place to live. They're just losing their homes, and they're having to go to the nearby islands. There's a wonderful thing that Brian's involved in, is you know, looking for no place without a mountain. That's what they're trying to look for, higher ground in order to survive. So yes, we do see the signature, both historical and present day. Wow.
Yeah, it's unfortunate we cannot seem to escape the effects of current climate change. Interesting, though, that you see the historical climate change that happens over a much longer period than it has happened in the Anthropocene now. But you're effectively seeing a museum down there as well with historical coral reef ledges that are being built, and it must be pretty cool to feel like you're going in a time machine as you descend down to the depths, working depths you're after.
Yes, it's like a TARDIS.
Very cool. Well, I think we're wrapping up now with the questions. Thank you so much, Sonia. That was an amazing talk. We've been tweeting about it. Follow us on Ocean First Education there to catch some screen grabs of this talk. We will be putting this up on the internet on our website, so you can share it with your friends or take another half hour of your day and watch it during lunch. And thank you, Sonia, for your time and for your amazing work at some of the most dangerous diving depths one can go to, and all the great work you do there. So thank you very much.
Thank you. Thank you very much.
All right, we'll wrap it up there. Thanks, everybody.